Okay, we're going to have a look then at how genes actually code for proteins, some of the uses of proteins and how they actually work. Now this is one of those sort of sections that some students do struggle with a little bit, so I am going to sort of take it quite slowly. Now don't forget that genes are in chromosomes, so they cannot leave the nucleus, and the copies of a gene, they pass out of the nucleus and go to the ribosomes where the proteins are assembled. And each gene or section of DNA has a sequence of DNA bases in it, and as we said before, there are 4 bases. A always goes with T, C always goes with G. So really the way I sort of try to describe this is, if you've got the nucleus, that's a bit like the library. Your DNA is like your reference section of the library. It's not allowed to leave the library because that is all of the instructions for what makes you. So what we need to do is we basically need to be able to take the code from the nucleus and take it outside of the nucleus to where the ribosomes are, so that they can then make the proteins exactly the same as the code says inside the nucleus. So there's a little process that has to take place. So I'm just going to talk through that now. Okay. So, as you know, the 4 bases are called A, T, G and C. Like I said, you don't need to know the names, you just need to know A and T go together, G and C go together. And this is known as a complementary base pairing. Okay, the base pairs form a code, and they are read in groups of 3 or base triplets. Each triplet specifies a particular amino acid, and if you look back at the last slide, this is what I'm on about when I say about a triplet code. Okay, it takes 3. So for instance, this one, A, T and G, that's a code. C, T and A, that's a code. G, G and C, that's a code. So if we take that then, so we've got our base triplets. So each triplet specifies a particular amino acid. Okay, so like I said, so A, T and C would be for one amino acid, whereas A, C and T would be for a different amino acid. The sequence of the triplets of bases on a section of DNA specifies the sequence of amino acids in a protein, because amino acids make proteins. As you know, DNA always stays in the nucleus and proteins are assembled at the ribosomes in the cytoplasm of the cell. So in order to do this, we have to have another molecule to take the code out of the nucleus and to the ribosomes. This molecule is called mRNA, which means messenger RNA. It just carries the message. So it carries a copy of the coded instructions in the gene out of the nucleus. The instructions in the DNA are like a recipe that is in a book and you cannot take it out. The messenger RNA is like a photocopy of the recipe that you can take out of the library. The messenger RNA is a single-stranded molecule, and it is a copy of one strand of a length of DNA. In other words, it is a copy of a gene. The messenger RNA then leaves the nucleus and goes out to the ribosome. Here it governs how the amino acids are assembled into proteins. The amino acids which you get from eating and digesting food are assembled into long chains, and the sequence of amino acids in the protein governs how the protein will fold up into a particular shape. Each different type of protein has a specific shape, and this is how enzymes each fit just their own specific substrate molecule, and we're going to have a look at that in a little bit more detail in a moment. But before that, I just want to talk about some of the uses of proteins. First of all, collagen. Collagen, your skin, bones, cartilage, tendons, ligaments and walls of blood vessels contain a type of protein called collagen. Collagen makes up some of the structure of your body and it is called your structural protein. Insulin is a protein hormone made in the pancreas, travels in the bloodstream to the target organs, which are your muscles, and your liver, because that's where you because insulin is all to do with sugar. It regulates your blood sugar levels, and many other hormones are also proteins. Hemoglobin. You have hemoglobin in your red blood cells, and that carries oxygen from your lungs to your respiring cells. Hemoglobin is a carrier protein. So if you're asked for a structural protein, you're looking at collagen. If you're asked for a carrier protein, you're looking at hemoglobin. Now, enzymes are catalysts that control all of the chemical reactions in your cells and your digestive tract, and we're going to have a look at those now so you can see how they work. So if you look, here's a diagram then that's showing you a little bit about enzymes, because again, enzymes are they are a catalyst, and if you remember, a catalyst speeds up a reaction, but enzymes are made of proteins. You have these reactions taking place for photosynthesis, for respiration, for protein synthesis with the ribosomes. All of these things need enzymes to help them to work, as well as, like I said, all your digestive enzymes. Okay, so we're going to have a look then at the shape of the enzyme and how it actually works. Enzymes, like all proteins, are folded into a particular shape, and the shape of one particular area of the enzyme molecule, called the active site, is very important, and you can see that in the diagram on the top right hand corner. The substrate molecule fit into the active site, and this brings them together so they can form a bond, and this makes a bigger molecule. In some cases, a big substrate molecule fits into the active site and a bond breaks, so two smaller product molecules are made, and this is like, for instance, in digestion, because you want to break your products down so that they can be small enough to be absorbed across the small intestines. So sometimes they join them together and sometimes they break them down. Now if you look on the left hand side, you've got two graphs there that show you about optimum conditions for enzymes to work. Firstly, if you look at the one on the top left, you'll see that pepsin works at a very low pH and likes that, whereas trypsin doesn't. So trypsin has to be at a high pH, so it's more on the neutral side. In the bottom left hand corner, you'll see a graph showing you the optimum temperature for enzymes to work, and if you notice, as soon as you get above 40 degrees your enzymes will stop working, and this is because they are starting to denature, okay, which means they are kind of unravelling. So please remember, don't say they're all dying off, you have to say they are denaturing and unravelling. So just to sum up then, proteins are made with the ribosomes in the cell. Proteins are important for collagen, insulin, hemoglobin and enzymes have a substrate, an active site that fits into the substrate, and they like to work in certain conditions.